Good morning, everyone. Glad to give a talk. So my background is quite different. I'm a chemical engineer and environmental scientist. So I'd love to talk about from a chemical engineer perspective, how can we utilize the dairy waste into something sustainable and valuable. And most of our other speakers are talk about the whey proteins, and I'm talk about the other 80% of the total solid in whey, the lactose. So when I talk about this process, I'd love to give a background on a particular whey. It's called acid whey you get from the Greek yogurt acid productions. So in the past few decades, there's a great switch to utilize people add more Greek yogurt in your daily life. And in 2023, we produce over 1 million tons of Greek yogurt, which lead to over 2.2 million tons of Greek yogurt acid whey as a byproduct. So it is quite different dairy with the stream because it is a high concentration organic waste you cannot dispose into environment. But compared to whey protein or cheese whey or sweet whey, the acid whey, it has less protein content, so you cannot utilize it to produce whey protein or other materials, and it has very high mineral content. You cannot just dispose in the farmland as fertilizers, and it is have very high the acid content, so it is not a very good feedstock for livestock. So in the past few 30 years or 40 years, many major Greek yogurt producers developed different methods to try to reduce the environmental impact or try to get some money from the acid whey. So they try to many process. They didn't reduce the environmental impact at all. They cannot make the project sustainable without federal funding, or they only utilize the small fractions of the acid whey. So what we did in our Huber groups, we developed an integrated process containing the member filtration and to remove separate the acid whey protein and minerals as a byproduct. And most important, we developed a catalytic-hydrolysis process to utilize the lactose to hydrolyze them into glucose and galactose. And after further purification and concentration, we produce a glucose-galactose syrup. It can be alternative sweeteners as high fructose syrup, high fructose cold syrup, and we sell it at 20 to 30 cents per pound. And in the past few years, we started with the benchtop scale batch system. We developed into a continuous flow reactors, and we scaled it up to a parallel scale continuous flow reactor with a treatment capacity of 1 gallon per minute. It sits in CDR right now, and it should be online very soon to produce GGS at very high scales. And we developed the fully economic analysis and LCA analysis to think, okay, this process can reduce a lot of environmental impact and it is profitable. But still, we are seeing the GGS at 20 to 30 cents per pound. Chemical engineers want to make something more valuable. So we look into the galactose and we find, okay, we can convert galactose into isomers called Tagatose. The reaction is very close to your isomerize the glucose into fructose to other sweeteners, and it was called a so-called low-calorie sweeteners because it has 92% of sweetness of sucrose, but only 38% of the calorie as sucrose and only 5% of a GI index compared to sucrose. So it is really nice for use the Tagatose for the diabetes and weight management, and actually it is really profitable because you can sell the Tagatose at $3 to $5 per pound. That is 10 to 20 times higher selling price. And unfortunately, Tagatose is very rare in the nature. You can only find them in the overheated milk or some plants in Western Africa. But about 100 years ago, people already find if you treat the galactose in bases, you can convert the galactose into tactose eventually. And yeah, so we find out the mechanism is just to isomerize the galactose into tactose in bases. And what we did is screen all types of the bases to find which kind of reagent or catalyst is the most effective. And we find out the calcium hydroxide, which is really cheap and abundant in the natural world. They should very good yield and selectivity towards to tactose. 80% conversion and 8% selectivity makes this seems like a really interesting catalyst or chemicals to convert galactose into tactose. So during our experiments, we mix the calcium hydroxide slurry with galactose. We form that very unique yellow color solid gel, and after we use acid to neutralize the gel, we find galactose. Most of the galactose convert into tactose, and we also find a small fraction of talose, which is also a very more expensive chemicals, but is very low content. So we developed the possible reaction mechanism, so it should have an equilibrium between those three types of sugar: galactose, glucose, galactose, tactose, and talose. And we are conducting the thermodynamic research about use DFT calculations and MD simulation, try to figure out what exactly reactions happen during the process. But that is more like chem's work. I am a chemical engineer, so what I care about is the reaction kinetics and the reaction engineering, so we can produce the expensive tactose in a quick, cheap, and simple way. So we conduct different reaction parameters on the effect of the tactile yield and selectivities. It turns out the reaction is really quick. You can have almost 80% the conversion within an hour. So it is a really quick process. So a reference, Bonimos, another company in Virginia, they use enzymatic process to produce tactose. They use 7-step enzymatic step. Production costs several days to produce tactose. We just need one step and within an hour. And the reaction just happens at room temperature. You don't need to heat the process, which is we put less energy. So this process is something you can produce in a continuously and at larger scales. And so those results come from the pure galactose. So the next step, we are thinking, can we just use the GGS with galactose and glucose a mixture to produce the tactose? So we isomerize the GGS with calcium hydroxide. It turns out you can eventually get a similar lactose conversion at 80%, but galactose conversion at 80%, but it takes longer time. And if you look at into the selectivity towards tactose, it dropped from 80% to 50%. So it's a sugar chemistry with when you involve the glucose, it's totally messed up. This is something we don't like. And if you look into the products, we not only have the galactose isomerized into tactose and talose, we also have the glucose isomerized into fructose. So the take point is if you co-isomerize the GGS, we will cost more calcium hydroxide, and those glucose will mess up competed with the galactose isomerization. So we better separate out the glucose first, then we run the galactose isomerization, then we separate the final products of tactose. So when we look into the different separation process, the most interesting part is called stimulating moving bed chromatography. That is commercialized technologies widely used in the pharmaceutical and petroleum and sugar industry, so it can effectively separate the different isomers and sugars Yeah, so the idea is because the sugars have different interactions with the chromatography resins. So if we play with the sugar solution direction and simulate it moving the bad position, so it will eventually separate out the glucose, galactose, and tactose. So this is a commercialized system, and we are working on this process to do this work. So we are working with a company called Orcam in Chicago to develop this technology applied in our lactose to GGS to tactose process. We have conducted the separation initialized preliminary test last year to treat 50 gallons of the GGS isomerization solution. And after the isomerization, we can have the tactose purity increase from about 30% to 80% in our first trial, and it is still an ongoing optimized process. And in working with Orcam, they will donate Huber Group a pilot scale GGS system in Babcock Hall in this year, and we can conduct the SMB separation of the GGS and tactose at pilot scale about 50 kilograms per week, and we are going to scale up the system. So in general, when we look into the mass balance, which is a very important concept for our chemical engineer, so if we put 1 ton of lactose at 40 cents per pound, we can produce around 400 kilogram tactose we can sell at $3 per pound. And we can sell about 500 kilograms of glucose and also about 500 kilograms of calcium carbonate as byproduct. So the whole process is really profitable if we start with the relatively expensive feedstock, the pure lactose, and the technology has been applied to Greek yogurt, acid whey, the cheese whey we get from Babcock Hall, and the milk permit, which is a lactose-rich stream we get from the lactose-free milk producers. Another thing is how can we use those lactose? Actually, we are going to produce the tactose sweetened chocolate milk in Babcock Hall in this summer, and we are also testing the tactose we produced in different types of other dessert like the gummy candy, cookies, and ice cream. So in conclusion, we are developing a sustainable and scalable neutrals process. So we use the American dairy waste, produce the tactose in American, and scale up the system to benefit the local American industry, agriculture industry, local agricultural communities, and benefit the health of American peoples. So that is what we have done in the whole process. Hopefully we can scale up the system and deliver the chocolate milk to UW people. And in the end, I particularly thank all these funding agents for my work, and particularly the CIS postdoc fellowship and from the DIH, and we are also working, thanks for our work partners, Orcam. So we are working, developed the technology to separate tactose.
Do we have one quick question? Oh,
there. What caught my attention was the sterculia tagatose assemblage. Have you, in your experience, come across any links as far as neurodegeneration?
That is a very good question. For now, we haven't done that part yet. So we are mostly focused on the produce the tactose in a cheap and quick way, because there's bunch at only one company commercialized to produce the tactose. They have done a lot of tests on that issues, and it is FDA generally considered as safe.